Good afternoon, everyone. I am Pei Ping from UCLA on behalf of Dr. Carol Watson, my co-lead and PI of the Skills and Workforce Development Core of the NIH Bridge to AI program. We're honored to present you Professor Fei Wang from Computer Science, UCLA School of Engineering, course instructor and director for the SWD Education Lecture Series 2022 to 2023, Fundamental Concepts to AI and ML module. Professor Fei Wang.
Thank you, Pei. Today, we are honored to have Professor Fei Wang to give a lecture on multimodal learning. Professor Wang is currently an associate professor of health informatics in Department of Population Health Science at Weill Cornell Medicine. His research interest is machine learning and artificial intelligence in biomedicine. He is the founding director of the Institute of AI for Digital Health. Dr. Wang is an elected fellow of AMIA and ACMI and a distinguished member of ACM. In this lecture, he will give an overview of methodologies of multimodal machine learning and how they can be applied in biomedicine. Without further delaying, I'm going to hand over to Professor Fei Wang to start his lecture.
Thank you, Dr. Wang. Let me share my screen. So can everybody see my screen?
Yes.
Okay, great. Thank you. It's a great pleasure to have this opportunity of presenting here. And the topic today, as Dr. Wang introduces, multimodal machine learning in biomedicine, certainly an important topic. And this is an outline. I'm going to start with an introduction, and then I'm going to talk about the different strategies of doing multimodal learning, including modality concatenation, modality fusion, similarity fusion, co-training, modality generation, and patchwork learning. And a lot of them, actually, I gave the name myself, so you will know what do I mean. So first, let's start with introduction. I think I don't think I need to emphasize more on what is multimodal learning and why it is needed. And I just want to cite the previous NIH director, Francis Collins, when he was introducing this precision medicine initiative and this New England piece, he explained that this initiative will really encourage and support, the research, analyzing a wide range of biomedical information, including molecular, genomics, cellular, clinical, behavioral, physiological, and environmental factors, which means that in order to achieve precision medicine, it is really important to integrate all those biomedical information from different scales and different types all together to better understand the individualized health status, and then come up with a tailored or customized treatment or management plan. So by multimodal learning, we really mean like those, different information types. Like I just show some examples here. Around every patient, you're going to have the electronic health records, you have the structured information, unstructured information, you have the physiological signals, you have videos, you have dialogue, you have biomedical literature, you have medical imaging, you have pharmaceutical research and development, and so on and so forth. All those data are related to each person's health status. And the question is, how can we perform integrative analysis across all of them? And each of these different types of medical information is typically referred to as a modality. And certainly, as you can see, different information modality have very different characteristics, and it is not that straightforward if we just want to combine them all together to perform some joint analysis. So multimodal learning is actually I would say, pretty fundamental concept in machine learning, and it is not something new emerged since precision medicine. Actually, when I did my PhD study like 15 years ago, we are doing something called multi-view learning. In machine learning, people sometimes also call each feature modality a view. So multi-view learning, people called it in the past, and now multi-modal learning, pretty much the same thing. And with the kinds of like the emergence and popularity of deep learning, people have been trying to categorize those different types of multi-modal learning research. So I'm just showing you one example or one type of way to categorize that, depending on because essentially you can think of it as how you want to fuse the information from different modalities. You can fuse them at the very early, let's say if you have a deep architecture like this, right? So you can fuse them at very early stage, you can fuse them in the intermediate stage, you can fuse them in the final stage. So people kinds of like call them like early fusion, intermediate fusion and late fusion. But I'm just thinking that this is not really informative for the practitioners or like our audience who really want to practice or either try out or develop multimodal learning algorithm yourself, right? This is certainly a good taxonomy, but may not be that informative if we want to really do some practice. So I'm just trying to kind of like, as you can see, in my categorization, I have many more categories, more than 3, certainly. But I'm just trying to summarize, like for these existing works, how they fuse different information modalities. I think that's a key because, otherwise, if you successfully fuse all those modalities, you just do whatever you do for single modality machine learning, right? So I think I'm going to talk about them one by one, so you can see what people have been developed. So first is modality concatenation, as you can imagine from its name. So it's just a simple concatenation of different modalities. Let's say in this case, it is a collaboration work of my group with clinicians from Hospital for Special Surgery. They have one of the best orthopedics medicine, in the nation. I think they ranked #1 for like a period of I think over 15 years. But in this case, it's really we are trying to discriminate osteoarthritis and rheumatoid arthritis. So these two are different types of arthritis, and patients with these two different types of arthritis, they in terms of phenotypical demonstration, there are lots of similarities, like they have like joint swelling, warmth, and diffusion. If we can like discriminate them well, then you can have a more tailored treatment, so that there is a better chance that you can intervene the patients early and give appropriate treatment options. The reason why this is important is because both types of arthritis gonna do severe damage to the joint, and a lot of times it's going to result in total knee replacement. So that's pretty painful for the patients. And in this case, as I said, so from pure phenotypical data, it is not easy to discriminate the two types of arthritis. So people have been using like pathology slides from the synovium. And so here, what we trying to do is, the reason why I call it multimodal is because we actually use two type of features extracted from the pathology slides. Number one is the expert-derived features. There are in total 14 of them from the human pathologist. And number two is because of the advancement of computer vision techniques in recent years. We also have a pipeline to kind of like extract the computer vision driven features. That's the cell density from those pathology slides. And certainly these two type of information are of you can view them as a different information modality because they are extracted very differently. And what we do is just to concatenate all of them instead of treating, processing them separately, just to concatenate both the human pathologist extracted features and also the computer vision extracted features and train a random forest and see what is the performance. And actually the performance is pretty good. The random forest achieve a performance, the classification AUC of around 0.91. And these are kinds of like the individual contributions of those individual features. And we rank them according to the importance score returned by the random forest. And with respect to each feature, we can also train a decision staff so that you can also see just based on a single feature. So what is what is the classification performance a single feature can achieve? So that's a macro AUC. What does that mean? And as you can see, although some of the features, especially its computer vision feature, the automated cell density, can do a pretty good job achieving an AUC of 0.88, but adding up all of them together, concatenating all of them together, we can still achieve improvement. That's what I said. Altogether, they can achieve an AUC of about 0.91. So this is one example where we don't really do anything special. We just concatenate all the features, with some simple normalization, z-score type of stuff, and then train the model on top of that. And there are also other examples, like instead of just concatenating those almost raw features, so they first do some pre-processing because some of the information modalities are more heterogeneous than like the example I just provided. So in this case, we look at the mimic data. That's, if you know what is mimic, that's the ICU data. And you have like structured information in tabular format. You have the physiological signals in continuous time series. You have the clinical notes in natural language. And you also have the chest X-rays that's available in Mimic4. So if people wants to because ICU is super complicated, some deadly event like sepsis, like death, like hypotension, all those kind of things could be due to very different causes. So you never know. Which feature modality plays a more important role in which kind of outcome prediction, right? So what these people do, actually, this is a team from MIT and Harvard. So what they do is still they don't do anything really complicated, but they pre-process each different type of feature or each modality of features in the different pre-processing pipeline. Like tabular features is just to do whatever I just introduced for the OA versus RA paper. They just do some simple normalization for those features. In the time series, they do a sequential model pre-training to embed them into some vector space. And for natural language, they do some transformer or pre-trained language model processing to embed them into some vector space. And medical image, they use pre-trained convolutional neural nets to embed them into a vector space. And of course, there are other features they use. Other dedicatedly designed architecture to embed those features into some vector space first, and then they rather than concatenate raw features, they concatenate those embeddings and then do whatever the classifications. And so as you can see, they still didn't really explore like the interactions or the associations across different information modalities. You're going to see in the later, in later part of this lecture, but now it's still, different modalities processed differently using different models dedicatedly designed for this particular type of modality, and then concatenate them. And this is a performance they have been predicting, I think, 10 or so different outcomes, including some pathology diagnosis, like is there long lesions, is there fractures, is there long opacities, pneumothorax, and so on and so forth. And they also predict like the length of stay and 48-hour mortality. So that's those, as you can see on the different columns of those heat map, and on the rows, that's these modalities means those different information modalities as I highlighted here. And within each information modality, like imaging, so they have from 4 to 10, those features are from 3 to 9, those are the different variables corresponding to the imaging features, and from 4 to 10, those are the modalities from the nodes or the unstructured text features, and these color actually encoded like with respect to a particular outcome, the prediction of a particular outcome. What if you use all different kinds of shuffling or permutations of, let's say you use 5 variables from 3 modalities. You just shuffle all possible combination of 5 variables from 3 modalities and see how much AUC you can get. So that's how you read this. So you can see that lighter means higher AUC, darker means lower AUC. So the more information, variable, and modalities you use, the higher AUC you can achieve, you tend to achieve. And this is a universal kind of observation, doesn't matter which outcome you are predicting. And at the same time, they also checked the stability of their prediction. So it seems when you use more information, so your prediction standard deviation also reduces. So that's a desired characteristic we want, because we don't want the prediction variance to be too large, which suggests there is a low level of confidence actually. So this is 2 examples that give you like simple modality concatenation. Either you just concatenate the raw features, or you concatenate the different feature modalities after pre-processing within each modality. And then you can think of, one of the limitations is you didn't really explore the interactions across those different feature modalities. And then let's see, what people have done when they want to explore the relationship or association across different information modalities, okay? So the earliest, not the earliest, but one of the classic technique, certainly you can think of, if you want to explore the associations across different information modalities, we should from simple to complex, right? So certainly by simple, we mean linear models. So like one of the classical technique that can achieve this goal, to learn the association across different information modalities. That's canonical correlation analysis. Let's say if you have 2 information modalities, that's XA and XB, so 2 different set of variables characterize the same data objects, and then you want to explore their associations. of what canonical, correlation analysis, or CCA, wants to do is to find 2 linear mappings. So one is with respect to feature modality A, one is with respect to feature modality B, and you want actually after the linear mapping, so in the mapped space, those vectors have the highest correlation. So we call this mapped factor, this ZA and ZB, the canonical factors, and their association, can be measured by this, which is essentially the cosine similarity between these 2 vectors. And what CCA tries to do is to find the WA and WB to maximize such an association. And actually, CCA is pretty popular in a lot of biomedical applications, and this is just one example. In the multi-omics space, where, let's say, these are kinds of like 2 different omics space. Let's say one could be like RNA seq, one could be the proteomics, and you want to leverage both information modality. And the first thing you are going to do is to map them into the same feature space, so that they are compatible with each other, and then you do whatever clustering or classification in that unified embedding space. Here, the embedding is linear after linear mapping, so it is a pretty classical. So I mean, they just run the CCA. I mean, typically people run add further when they maximize this association or correlation. They further add a sparsity regularizer like the one norm of WA and WB to further enhance the interpretation, because in that way you can see, I mean, for the different information modalities, which variable really contributes, because it can help shrink the not relevant variables coefficient to 0, right? So that can further show you like, or pops out like which the contributing variables. And here is essentially what they do. And after they do the mapping, they kind of like do the clustering on the embedding space and derive the four, different subtypes or cell clusters. In this case, it is the ovarian cancer subtypes. I mean, not the cell clusters, but patient clusters as the cancer subtypes. And these OC1, 2, 3, 4 are the data. I mean, the clusters or subtypes derived from this data-driven approach, and this rows are the labels that are given by the oncologist. So you can think of these as a gold label, and these are the derived label, and they check the association, as you can see. So darker color means high association. So this, I mean, by combining, these two modalities, you can recover these ovarian cancer subtypes pretty well. And they also do a classification using these as a gold label, and then do the subtype, classification. And they compare, like, what if you just use, I'm sorry, it's not RNA and proteomics, it's miRNA and mRNA. So what if you use them individually, and what if you use them jointly? The joint feature is derived by the sparse CCA mapping. So as you can see, so this using both modalities always can lead to better classification performance. So this is one of the I think classical way of like linear way of learning, exploring associations across different information modalities. And actually CCA has been used in more recent studies as well. Like this is an example when oncologists or rheumatologists, they want to study the disease activities. They want to combine both the genomic data and also the histology slides. So this is one paper. What they do is so first, for every patient, you have a set of cells extracted from a particular tissue extracted from the patients. I mean, depends on different diseases. In this case, they study cancer, so it could be from tumor. But for like rheumatology, it could be from synovial. You do sequencing, so you can get the genomic information, but you can also do this histology, so you can get their pathology slides, and they want to check the associations between the genomic information and whatever that is demonstrated on the pathology slide. So you can see for certain genes, if there is any disease activity demonstrated on the pathology slides, let's say a certain type of concentration of a particular type of cells. So it can explain, like, if there is a particular type of gene that can, lead to the different disease activity. So that's kind of like the motivation. And here, so they just use CNN, I mean, convolutional neural net pipeline to process the pathology slides. It has nothing to do with genomic information. And after they extracted the histology slides, information as a vector, they do sparse CCA to explore the association between the features extracted or the vectors extracted from the histology slides and the gene, RNA expression data. So you do a CCA, you can find like a linear direction where a kind of like a transformation or linear mapping of your histology slice feature and your gene expression feature can be maximally correlated. And here they just show some examples. This one is a breast cancer. This one is a brain cancer, and they show like the top one. So the first, if you rank the canonical value after you do CCA from largest to the smallest. So this is the largest canonical value. They are corresponding canonical vectors. So this is a vector on the histology slice side, and this is a canonical factor resulted from the genomic side. So you can see like this is a factor with a high value where you can see a very high density of malignant cells, and this is a factor with low value, which corresponding to like like muscle tissues. And this is a kind of like the different genes that got enriched in the corresponding genetic factor. So in this way, so you can kind of like see the top component was enriched for genes involved like in cell adhesion and also in the extracellular and molecular functions related to the ion channel binding and all these kind of activities. And images with the high magnitude positive values have well differentiated nuclei and the minimal extracellular connective tissue, but for the low value it has a high magnitude of extracellular connective tissue. So it can explain some of the potential mechanism. But still, in this work, they also kind of like did not do anything really complicated, but really using CCA to explore the linear associations across those different modalities, although they pre-process the imaging part first. And another work I want to mention is one of my collaboration with Dr. Yuan Luo's team from Northwestern, where we still use the linear method, but in this case, it's not CCA, but we have two type of information for patients with hypertension. So one is a phenotypical variables, one is a genetic variables, and we do something called the joint matrix factorization. So we hope to embed the two matrices because they have a shared modality, a shared dimension, which is the patients, and we hope by doing joint factorization we can have a patient group factor and also a phenotypic group and also genetic groups derived, and so that we can kind of like see the correspondence between the phenotype group and the gene group through this joint matrix factorization. But no matter what, this is still a linear approach, but clearly we are trying to blend the two modalities in CCA as well, blend them in a linear way. And then, with the boom of deep learning, certainly people are, thinking of rather than, as you can see, so for CCA or, matrix factorization, you essentially embed your raw, information modalities into a unified space and do whatever you want to do in that unified embedding space. So we can certainly do that in a nonlinear way. So this is a work from my group. So we have done like using the DTI diffusion tensor imaging to classify the patient is more likely to have a Parkinson's disease or no. So there are different ways of quantifying the DTI images. So the different ways of quantification can be viewed as a different feature modality, and we are kind of like treating those different feature modalities. We first use this free software to transform those brain images into a brain network. So you can think of as a graph. And then we do graph convolution to extract features as a vector from each quantification of the DTI brain network. And then we do max pooling to kind of like combine all of those different features from different quantifications as different modalities, and then do the classification. And this is the result, as you can see. So these are the different ways of doing the quantification for DTI images, and these are the different ways you do feature preprocessing. You can just use the raw feature. You can use PCA, fully convolutional nets, and two-layer of fully convolutional nets, and graph convolutional nets, as you can see. So with respect to each view, the graph neural network can achieve better performance than if you further combine all of them, so you can further improve the performance. So that's not surprising. And in this case, so we kind of like using a nonlinear methods. It's the graph convolution nets to, of course, our input data are in forms of graphs to extract the features into vectors, and then we do max pooling to do kind of like to combine the embeddings or the extracted features from GCN, and then do classification on top of that. Another work I want to mention here is, we have looked at like the images, like the genetics, and another thing we have been worked a lot, especially in my group, is electronic health records. And in the electronic health records, so there are naturally two different feature information modalities. One is a structured information like the diagnosis code, procedure code, medication code, whatever. And the other is unstructured information, that's the clinical notes. So we kind of like trying to combine both information modalities to do a prediction. In this case, is the prediction of the risk of onset of acute kidney injury. And here we use a network called, memory network. I mean, this is several years ago, of course. Now we can all use transformer type of architecture. But in this case, we kind of like use a memory network, and the way how we combine them is we use a hierarchical LSTM to extract the information or embeddings from the clinical note sequence for every patient, because the patient gonna have multiple visit, and every visit you're gonna have a note or a set of notes. So if you can consider the different visits, is going to be a sequence of nodes. So that's why we use a hierarchical LSTM to embed the set of nodes to a vector, and we use this vector as a query. So this memory network is initially designed for information retrieval purpose. So you have a query, and you have a set of potential answers in your memory, and you match your query with your answer, and you retrieve the right answer. Here we just use the structured information over every visit as the kind of like the potential answer or matched data in the memory, and we match the nodes, the embedding from the nodes with the historical structured information. And because we believe, so there could be like a certain thing that got highlighted, like the conditions or procedures, certain events in the past that can enhance or highlight your risk of AKI in the future. So we combine them and do the prediction, and we certainly minimize the prediction loss. And we just show this is two different prediction horizons. So one is 7 days, one is 14 days. So essentially, we can, what we show is using this combination technique. Of course, we can use different kinds of classifiers, but using this combination technique, we can always achieve better performance than using either of the single modalities. We kind of like used adopted this architecture in another work, which is if you remember, I showed you the classification Parkinson's disease context. So what if we combine the brain image with the patient clinical records, right? So here, I mean, as you can see, this is still a memory network architecture. The difference is we don't use, clinical notes here as a query. We use the brain network here, and the feature extracted from GCN as a query. We still, put the structured visits in the memory, and we combine them and do the classification. And we find that if we combine them all together, it can do better than you just use either of them individually. So these are just kind of like some examples on, how can we do modality fusion from linear, like CCA matrix factorization, to nonlinear. This nonlinear could be in a more implicit way, like you can embed them and then do like some sort of concatenation, like the multi-view GCN work, or you can do this more implicit way, like you further blend them in the memory network. But you certainly you can do it in other ways as well. And just showing you the different ways you can explore this modality fusion. Another type of way people do, like when fusing different information modalities, is rather than just fuse the modalities, where essentially the principle is you have to embed those modalities into some embedding space. So people have also been thinking of what if we can build similarities according to different modalities, and then rather than fuse the modalities, let's fuse the similarities. So first, before that, I want to introduce like how do you construct it or calculate similarity, right? So certainly if your data is available as a network, in other words, like you already have similarities encoded in your data, then you don't need to worry about this. But a lot of times, as like I just follow the same setup. So what we have are the feature vectors. So typically what people do is that, like in this case, you first calculate Euclidean distance, and then you translate the Euclidean distance to a similarity by a Gaussian trick. And the width of the Gaussian defines the locality of your similarity. In other words, if your sigma is large, so the locality you care about is also large. If a sigma is small, so the locality you care about is small. So by large and small, I mean how fast your Gaussian is going to decay to zero, right? And then one of the things I want to mention here is in one of my early paper, we studied like the impact of the sigma. It can actually impact your result a lot. So like in this case, we look at this 2 half moon data set. Ideally, you're going to have the top half moon as one class and bottom half moon as another class, and you can correctly get it when you set sigma to 0.15. But if you increase sigma to 0.25, you're going to have result like this, which is not surprising, because essentially you're increasing your sigma to be too large value, and which confuses your top half and bottom half moon. So setting up sigma is tricky. So people have been proposing, instead of using a unified sigma, so let's just use a sigma per each data pair. And this sigma ij is calculated in this way, which integrates the locality information or the neighborhood information with respect to each of the data point in your data pair, right? So this is just the one way. I'm going to introduce this paper in the following slides, but this is this idea is kind of like proposed a long time ago. So with this individualized pairwise width, so it can kind of like consider the surrounding context of every data point. So it is more robust to different kinds of data distribution. But no, I mean these two slides are just showing you how we can translate the data vectors or information representations into similarities, and then let's look at how we integrate them. So this is one work published on Nature Method, so where people also look at the omics. One is mRNA, one is DNA methylation. So I mean, they first, calculate the patient. This is cancer patients. So the method is called SNF, similarity network fusion. So they first calculate patient similarity based on their mRNA data using this equation, okay? They also calculate the patient similarity according to the DNA methylation information, and you can construct the patient similarity, two patient similarity matrices, and then they somehow fuse them and come up with a unified fused patient similarity network, and then do spectral classifying on top of that. So the question is, how do they do the fusion? So it's a pretty interesting technique that is actually proposed in a computer science conference. They leverage them here, actually from the same set of authors. So this is kind of like the similarity calculation, and then they also this is P, as you can think of just as a normalized version, raw normalized version of similarity network, but they added some background noise to make sure the connectivity, so there's no isolated singletons in your network. And then they define this propagation network. As you can see, it's just related to the neighborhood of every data point. So to propagate the similarity across, the neighborhood by neighborhood, and so they propagate that in this way. So this is normalized similarity network. They multiply this network on both sides with this propagation network, and there is some convergence analysis showing that this is going to converge, and after it converges, the final fused similarity network is just the average of them, and you can extend this to more than two information modalities, and they kind of like tested this on 215 patients with glioblastoma multiforme brain cancer patients, and they have their information from mRNA expression, miRNA expression, and and the DNA methylation. So three different modalities, three different similarity matrix, networks. As you can see, individually, these lines showing the connectivities across different potential patient clusters. That's the subtypes of this cancer. So you can see there's still lots of connectivities that are going to confuse the subtyping process. But after the confusion, after the fusion, as you can see, so these three clusters are pretty well separated. So this is a Nature method paper, and another, as you can see, so this kind of blending, although it's mathematically, how it does, but after it comes, because this is going to proceed multiple steps. So after it converges, you actually don't know what is, how this final converged matrix is related, or how it is transformed from the original matrix. It is not that obvious, or it is not that interpretable. So another highly relevant topic is what the so-called multi-kernel learning in machine learning. So essentially, they call every similarity matrix a kernel. So instead of blending them in some complicated way that is hard to understand, so let's just do a convex combination. Like in this case, so we have Ki as the individual kernel matrix and G as your final matrix. We just do a convex combination. In this case, I wrote it in this is my paper. So I wrote it in a general format. So you just measure the discrepancy between your final kernel and each individual kernel, and you weigh them. And this combination is a convex combination, and you put some constraint. Like in this case, I want my final kernel matrix to be doubly stochastic. So that's a constraint I put. And so in this way, after you solve this problem, the alpha, the combination coefficient, can be actually viewed as how important each modality is. So it is more actually interpretable. So you can tell people. So it is I blend them according to like this modality contributes 0.6, this modality contributes 0.4. And my students have been applying my technique to example like I showed here to discover subtypes of Parkinson's disease according to the motor information and non-motor information. We identified 3 different clusters of PD patients, and these are just some interpretations according to different clinical assessment, and they make a lot of sense. And it got published on a pretty decent Parkinson's disease journal. So that's kind of like the third thing. About how do you want to fuse information? So through the similarity fusion. Another topic I want to talk about, which is not talked a lot about people nowadays, but in like 15 years ago, this is a very important work called co-training. So what co-training is, so you don't really transform the feature or the information modalities. You just treat them separately. And you train a classifier on modality 1, you train another classifier on modality 2, and let's say you also have a set of unlabeled samples, and then you use both classifier to classify those unlabeled samples, and both classifiers can tell you, like, some samples I have a high confidence it should belong to class 1, let's say. Another classifier says, I have a set of samples, I also have a high confidence, right? And then you add up those high confidence samples back to your original pool of labeled samples and like training samples, and then you redo these classification process again until some termination condition is satisfied. So in this way, you kind of like implicitly, blend the information from the 2 different modalities through the classification process. And there are lots of this paper was initially published on the theoretical computer science conference, and there are lots of mathematical proofs showing why this co-training type of framework can work. And I actually did a literature search. I did find this paper where they use, this is from IBM TJ Watson, they use co-training to blend structured and unstructured information of electronic health records to do some prediction of whether hypertension is controlled, not controlled, or uncertain. So they essentially show that with this co-training technique, you can get better performance. So I just want to bring this to people's attention, so you are aware of there is this kind of a technique. It can be like applied to like the scenarios like, what if I first embed my features into different embedding space and then apply this co-training technique? It can work as well. It is more like a technique for implicitly combine or blend your information across different modalities. Okay. And then, so I think lastly, patchwork learning is kind of like a future direction, but modality generation. So I initially don't have that, but I thought, we are in the AIGC era, so we cannot, avoid talking about AI generated content. So certainly this is a little bit different on blending, but really, what if I have one modality? Can I predict another modality, right? So this is modality generation, and one of the work from James So from Stanford is this Babel architecture, where certainly it is a generative model. You want to do modality generation, so it has to be a generative model. It is actually, in this case, is the RNA, 2 information modalities, RNA and ATAC, and they kind of like build a hybrid variational autoencoder, so that RNA and ATAC, they have own encoder decoders, but they also have blend decoders. So the blend decoder means that the RNA encoded vector can also be decoded to ATAC, so that you can generate, let's say, if you're only given RNA, it can generate ATAC, and vice versa, right? So I will skip this is a basic a slide showing what is a variational autoencoder. So it is, you just need to remember it is essentially a generative technique. So given the input, you can generate the output. So this is how did they show, like, you know, this is kind of like expert labeled, like cell types and their UMAP embeddings, and this is a kind of like you know, just a given one modality. I forgot, this should be given the mRNA, the gene expression modality, and they generate the other modality. I think probably this is given the RNA-seq modality, the generate ATAC modality, and then they kind of like generate these UMAP embeddings, and they kind of like showing the validity of their generated modality. As you see, I mean, these different cell clusters or cell types, they pretty much existed there, and the geometric relationship still preserves. So this is one example, and we have a recent paper. So we kind of like designed a more complicated model based on topic modeling. So topic modeling, if you know, is also a generative model. So we, in this case, look at three modalities. That's RNA expression, the ATAC peaks, and also the proteomics. And we also we call it imputed modality. So from like the proteomics, I mean from the RNA expression to impute the proteomics, or from the proteomics, I mean from the RNA expression to impute the ATAC peaks. As you can see, the patterns. So this is a ground truth. This is a ground truth, and this is a generated modality. It's pretty close. We also have some numerical results, and so I'm just showing you this is possible. And the last thing I want to talk about is another way of generating the information modalities, right? It's through knowledge graph, and this is excellent work from UCSF. And so as you can see, so they have a knowledge graph, and the problem they are trying to solve is from the UCSF EHR cohort. Certainly, you only have the electronic health records or the clinical information. We extract a set of clinical concept, let's say the diagnosis or medications that frequently appear in this cohort. So can you find the potential genetic mechanisms like for this cohort? Okay, so essentially what they do is they first map those clinical concept into the knowledge graph, and then they do random walk, so that for all the rest of the nodes, you can have a probability like how they are associated with these phenotypic nodes. So you can, so with respect to all those genetic nodes, so you can rank them according to the relevance after random walk, and that can give you some sense on what could be the genetic cause or mechanism of this disease. In this case, they look at the obesity. So I mean, this is kind of like the relevant. You can, because when you do the propagation, you can also look at the other phenotypic variables like the diagnosis. These are the most relevant diagnosis variables, and as you can see, so this is a particular gene that is known to be associated with obesity, and so as you can see, this is kind of like a rank of this gene in different groups. As you can see, in the obese group, it is ranked pretty high. So that shows that this knowledge graph can really find out the valid genetic cause of a particular disease. I mean, at least the potential. And they also check the overlap of the gene list could be related to obese in the GWAS study. And so this is kind of like the number of genes they can find to show the validity. And I think this is a knowledge graph that is built in my lab, and I think I think I can still show you this. We have a small video, so just in case people are interested about our knowledge graph.
Biomedicine has an enormous volume of highly specialized knowledge accumulated from various sources. Here we present the IBKH, a knowledge base which has harmonized data from 18 resources. Let's say you're interested in finding novel treatments for Alzheimer's disease. You can begin by generating a first neighborhood subgraph of Alzheimer's using the Entity Query Search tab. Note that there are multiple entries for Alzheimer's, which are indicative of different types, which you can learn more about by hovering over the information icon. Right now, we're looking at the top 200 connections, but this can be changed via the limit. To find drugs that currently treat Alzheimer's, let's go to the Path Query tab. We input Alzheimer's and a relation of treating drug and add it to our path pool. You can add more than one path to this pool simultaneously. You can then visualize the drugs that have direct connections to Alzheimer's. At the top, you can find the cipher query that lets you retrieve data from the graph database. You now want to find novel drugs that could treat Alzheimer's. Our knowledge discovery module uses graph embeddings to predict the most relevant entities to the input. Let's input Alzheimer's and submit. Looking at the drug section, we can see that sodium bicarbonate is a link predicted by the model. By looking at the evidence, we can see the path between the drug and Alzheimer's. Finally, you want to explore some characteristics of a cohort of Alzheimer's patients who have TREM2 mutations. The IBKH allows users to input multiple entities describing that cohort. Our model will predict knowledge in the IBKH that is associated with the inputted cohort and the evidence for those associations. All in all, the IBKH provides innumerable ways for users.
I'm sorry. Again, I mean, this is a knowledge graph. If you're interested, I mean, build in my lab. If you're interested, you can reach out to me. But it is, I mean, the paper was just accepted. It's pretty powerful. I think the last thing I want to mention is more like a future direction. So what we have talked about now is multimodal learning or multi-modal learning is more like this. So you have like data objects, patients, you know, you have different information modalities. How can you fuse them to do some analytics? And there is a related topics is on the other side. So you have this, I mean, one shared information modality, but you have patients from different institutions that you cannot really share information because of privacy concerns. So how can you jointly learn a model in a privacy preserving way? But what I mean by patchwork learning is really, I think a lot of times you have this situation where you have a different data set, where each data set you have different information modalities, and these information modalities may not be shared across different cohorts. So in this case, you can think of it's extension of multimodal learning in a multiple data set scenario. So it is a challenge in a sense that across data set you may have shift. So how can you do that? This is one of the things I want to call people's attention, and we have done some recent work on this, and we are happy to communicate if you're interested. And I think that's all I want to talk about. I think it's almost 4. Thank you.
Thank you very much, Professor Wang. So it's a very inspiring talk. So, I'm sure many of us have learned a lot. Clearly, myself have learned a lot. And if for the audience, please enter your question under the Q&A. We have a few minutes for questions for Professor Wang. So I'm sure other people are maybe typing in their questions. So meanwhile, maybe I have a question, right? So recently we have witnessed all these, breakthroughs, right? So in AI field, everybody talk about, right? So how would you view that may help assist in this multimodal learning in this biomedical field, right? Like, for example, things like GPT and the related things about there's like image generation. So it seems that at least in other domain, in the general domain, right, it has delivered quite amazing result. Do you envision that can be integrate, into this, machine learning models and systems used for medicine and health related area?
Yeah, that's a great question. That's why I said, I mean, I need to talk about AI generated content. So certainly there are a lot of potentials, but I would say we should be cautious about the actual scenarios it can be used. For example, people have been, since it passed the, doctor's test. So, can we use that in clinical decision support? So we have to pay attention to that because, the faithfulness we call, like, or the factuality of the content generated by those models are not guaranteed. But like in the case I mentioned in this talk, like multimodal learning, if the goal was to predict, another modality or to augment certain, modalities to improve prediction, that is certainly possible. So I do think there are lots of potentials, but we need to really think about the actual scenarios where it can be used.
Thank you. So now I see 3 questions. The first one is, can you elaborate on the intuition for patchwork learning? And can you provide a reference for it?
A good question. So you're going to see a reference probably in a month. Okay. So actually, we invented the term, and we are writing a perspective introducing this. So the intuition is pretty obvious because we talk about federated learning, we talk about multimodal learning, but in reality, it's typically like I have one data set including genetics and imaging. I have another data set including EHRs, but how can you do joint learning across those 2 different data sets with different information modalities? Unfortunately, that's the truth in a lot of the disease area. So that's how, we got, motivated to propose like this kind of setup. I'm looking at another question. So interested in patchwork learning.
Right, there's a lot of interest in the patchwork learning, and so another question is like to learn more like how to compare characteristics, right? For
yeah. So you also touched the pinpoint. So essentially, the challenge, if you will, call from patchwork learning compared to multimodal learning is the cross data set shift. So, how do you kind of like compare and align? So, domain adaptation or whatever, transfer learning has been talked a lot, but if you further consider the missing modalities of the features, there are certainly a lot of methodological challenges. So we can, if you're interested, you can send me email. We can talk offline about more details.
Thank you. So since we're running out of time, there's one last question we would like to answer. So is there best or state-of-art method for how to combine features? For example, concatenated features or embeddings, or do you just have to empirically test them in each use case?
The answer is the latter. So there is no like golden rule, like this is the method you should use. So people are still developing new method, and not surprising because the new technologies also are rapidly developing on like capturing new modalities, right? And like you said, so we have to really test them, and not just with respect to different modalities, but also with respect to different data sets.
Thank you so much. There's no more question. Thank you for the excellent lecture. So, and thank you so much. And thank you for all the audience. I'll hand back over to Dr. Pei Ping.
Thank you, Professor Fei Wang. What an inspirational lecture. We deeply appreciate. I think our audience appreciate. You could say we have had an incredible turnout today, and many of them are clinicians, and many of them are in basic biomedical research. And we appreciate you've left your contact info for us, and we will be contacting you on these very innovative concepts. Me, myself, personally, also thought it's brilliant that you come up with the concept of patch learning.
Thank you.
Love to hear more from you. Good afternoon again, until next week, Professor Wei Wang. And thanks again, Dr. Wei Wang, from all of our participants and audience. Bye-bye.
Thank you.
Bye-bye.